Hello students, my name is Vineet Kumar Singh, Assistant Professor, School of Pharmacy, BBD University, Lucknow. Today's topic of discussion is beta-lactam antibiotics. So, firstly, we have seen the basic things about beta-lactams, then go one by one their different derivative of drugs. So, beta-lactam uh, drugs, it's why is name beta-lactam? The beta-lactam is that mean key in the main core structure of their uh, drug. They have a beta-lactam ring. As you see here, this is a beta-lactam ring. Yes, either penicillin or cephalosporin, all all beta-lactam antibiotics contain beta-lactam ring. That beta-lactam ring is the pharmacophore and the ultimate pharmacological action is due to this ring. So all the compound which have a beta-lactam is called beta-lactam antibiotics. So first the compound in this class is penicillin. Penicillin is the first one, a naturally occurring drug which is known as beta-lactam antibiotic. So basically we know that the penicillin is a very famous things, very famous antibiotics from last 50 years or more. So how it discovered the first beta-lactam drug? How discovered? So it is discovered in 1928 by Alexander Fleming. Actually, in the lab, while working in his lab, trying to kill a deadly bacteria, he noticed a blue mold growing on dish. Learned that it was the mold Penicillium notatum. Penicillium is found in this mold, and they noticed that the bacteria around the mold was dissolving. Actually, Alexander Fleming and his colleagues worked. They are worked in lab, and actually they are working in some kind of bacteria, and they noticed that there is some kind of fungus growing, some kind of mold growth, and that's the name is Penicillium notatum. And where there is this fungus growth, there is a no bacteria. It means this Penicillium notatum mold kill all the deadly bacteria. Then they decided to extract out what is the main active constituent in this mold fungus. So the discovery of penicillin in 1928 and the final approval of drug in market is around 1940. It's very important drug in World War because wounded there is a lot of person wounded and get infection. So penicillin was the very very important drug that time. So Alexander Fleming does not extract out the active constituent from the mold. They spend remaining whole of life they working on, but unfortunately they cannot. But their assistant, there is two assistant, they done it, and finally uh, Alexander Fleming uh, also got the Nobel Prize for this. So for 9 years, nobody could verify the penicillin notatum to get the pure penicillin, as I told. Finally, in 1938, a team of Oxford University scientists, headed by Howard Florey and Ernest B. Chen, helped to develop penicillin. So, as we told that the Fleming, you know, uh, They tried much, but unfortunately they didn't extract out the penicillin. So penicillin is a naturally occurring drug, naturally occurring uh, antibiotic, which was not intentionally discovered by Fleming. It's unintentionally means sudden. There is a sudden discovery. They work for something else, but find this. And so initially it's a penicillin notatum, the source of penicillin. And here we look the structure, structure of penicillin. So the ring which is highlighted in red in color, that is called a lactam ring, beta lactam ring. This ring is contain oxygen molecule and nitrogen molecule. And here is this is a penicillin structure. In penicillin structure there is a beta lactam ring, there is a group R, there is a mention in R, that means there can be change happens. There is R group. Is a sulfur group. Sulfur group also is and five-member ring in penicillin, while in cephalosporin there is six-member ring apart from the beta-lactam ring. It also contains acid, COH, then methyl groups and also oxygen. This is a part of beta-lactam ring. It contains a beta-lactam ring in their molecular structure. Nitrogen is attached to the beta carbon relative to the carbonyl ring and hence the name. So before going further, we should note the what is the positive and negative gram bacteria, because their positive and negative characteristic, the rest of the antibiotic will act accordingly. So there is some basic changes like there is outer membrane, this is lipoproteins and a peptidoglycan chain. So in gram negative, we see that there is a peptidoglycan chain is thin, while in positive, the peptidoglycan chain is very thick. As we see, there is a very thick, and this thickness of peptidoglycan chain, it ultimately affect the mechanism of action of different antibiotics. So here the more precise structure like polysaccharide chain, peptidoglycogen. It's like this kind of structure. It's attached one by one structure. There is to remember the difference in the cell wall of gram positive and negative organism. Think some abbreviation, some kind of mnemonic signs. Yeah. So to learn that thing, so we just make some funda like think a boring long PowerPoint presentation. Long PPT will be your mnemonic guide. Long PPT. Suppose that I'm presenting a word through PPT na. So yeah, you can learn the long PPT, and their abbreviation is long for lipopolysaccharide, outer membrane, and for negative gram. While positive means peptidoglycan was thick or teichoic acid. So positive gram-positive bacteria have two main things: peptidoglycan chain that is very thick, and also teichoic acid. While in negative there is lipopolysaccharide outer membrane. Okay, so we Okay, so now the classification. Beta-lactam classification. The first one is, of course, penicillin. The second, cephalosporin. And third, other beta-lactam drugs. Other beta-lactam drugs contains like cefamycin, carbapenems, oxacephalosporins, beta-lactamase inhibitors, and monolactams. These are also beta-lactam antibiotics apart from that penicillin and cephalosporin classification. So beta-lactam structures, यहाँ हम देख सकते हैं यहाँ पे पेनिसिलिन स्ट्रक्चर ऑफ पेनिसिलिन, cephalosporins, carbapenem and monobactam. So all the structure have different, but only one thing is very common, that there is a beta-lactam ring. All the structure contain beta-lactam. Yeah, their substitution could be changed. But the pharmacophore is beta-lactam. Like there is 5-membered, 6-membered, and there is a also 5-membered ring. There is a also sulfur group attached to this. So penicillin structure, we can see that there is also R group, means there can be possibility of change. Then how do they work? The beta-lactam binds to penicillin's binding protein PBP. So these beta-lactam bind to a specific protein which called penicillin binding protein. So short name is PBP. This PBP is unable to cross-link peptidoglycan chain. The bacteria is unable to synthesize a stable cell wall. The bacteria is lynched. Up, that is a mechanism of action. The amide of beta-lactam ring is usually reactive to the ring strain and conformational arrangement, which does not allow the lone pair of the nitrogen to interact with the double bond of carbonyl. Beta-lactams acylated the hydroxyl group on the serine residue of penicillin binding protein active site in an irreversible manner. This reaction is further aided by the oxenium hole, which stabilizes the tetrahedral intermediate and thereby reduce the transition state energy. There is a one by one we discuss the how that mechanism of action of beta-lactam going on. So as we know that there is a lactam ring, beta-lactam ring. So first of this, what happened? The hydroxyl attack the amide and form a tetrahedral intermediate. This amide group, tetrahedral, this is attacks. The next, the tetrahedral intermediate collapse. The amide bond is broken and the nitrogen is reduced. And with that, let's go on to this side. This is broken and nitrogen will reduce. The protein binding, the penicillin binding protein is now covalently bounded by the drug and cannot perform the cross-linking action. The cross-linking action in peptidoglycan, as we earlier see that there is a structure of cross-linking of peptidoglycan chain. So basically the penicillin act on there. So क्या करता है वहाँ पे cannot perform the cross-linking action. then what will happen? Then the cell wall will finished. The cell wall will not remain intact, and ultimately the bacterial cell wall totally ruptured, and bacterial uh, inner material will not safe, and the bacteria will die. This is the structure penicillin. You this uh, this kind of presentation of hydrogen bond on other groups. This means that the showing the conformation that the plane of the hydrogen attached toward is something different. So here is acylamine side chain, beta-lactam ring, carboxylic acid side chain और ये thiazolidine ring. Thiazolidine ring and beta-lactam ring, and apart from this two more things, carboxylic acid and acylamino side chain. The next one is nomenclature. So nomenclature of penicillin is done to different system. So as we know that there is a CS system and USP system. USP system is United States Pharmacopoeia system, and a CS CAS it's chemical abstract system. These both two systems have decided that things. Okay, good. Now it's good. Okay, so according to abstract system, according to this system, penicillin are numbered starting from S, that is sulfur. Thiazolidine ring start from S atom. Sulfur atom is assigned the 1 position and nitrogen assigned the 4th. If we start from, where is my cursor? Okay, what the 1 minute. Okay, now. Okay, if we start from here, if Q to sulfur 1, then 1, then again 2, 3 and 4. So sulfur assigned 1 position, nitrogen is assigned 4th position, and is called 6-acylamino 2,2 dimethyl 3 carboxylic acid. What called 6-acylamino 2,2 dimethyl 3 carboxylic acid, and where United States Pharmacopoeia system. In this system, penicillin is reverse of CS system, just opposite. According to this system, nitrogen atom is given the position 1 and sulfur atom is assigned 4th, just opposite. Where in CS system, the sulfur got assigned 1, while in USP system, sulfur assigned 4th position. And then after this call is 4-thia-1 as a bicycloheptane. So now according to ISP, this ISP name is called 4-thia-1 as a bicycloheptane. Okay. So now the chemical degradation of penicillins. So penicillin degraded by the presence of acetic acid, a pH medium. So what's happened? Penicillin, alkaline, lactase, penicillinase. Lactamase or penicillinase. Penicillinase, it's a enzyme which invade the penicillin, which degrade or which help to degrade penicillin. So penicillinoic acid, then penicillinic acid, and second chain is also like this. So ultimately, action of lactamase or penicillin is what? They directly trying to hit the lactam ring, and they cleavage the lactam rings, open the lactam rings, and the compound will metabolize. And stereochemistry. So position 1 and 4 according to the name of CAS. It contains 3 chiral carbon atom C3, C5 and C6. So 3 chiral आइटम कौन कौन से हो गए? पहला तो C3 हो गया हमारा, उसके बाद C5, यहाँ पे एक हो गया C6 कार्ल का आइटम, वो जहाँ पे 4 डिफरेंट ग्रुप लगे हों कार्ल का. C6 L configuration, C3 and C6 chiral inters are trans to each other. All synthetic and semi-synthetic penicillin have same absolute configuration, that of natural 3s, 5s and 6r. 3s, 5s and 6r. That's the stereochemistry dimensions. 3s, 5s and 3r. 6r. So that would be this would be very important question. Will ask in many more competitive examination. The conformation of penicillin stereochemistry of S form or R form. So must be remember this. The next topic is the classification of penicillins on the basis of source, routine, route of administration, spectrum of activity, resistance to enzymes, resistance to acids. So there is a huge classification at all. So the natural penicillin G, while oral ampicillin are the thing. Then spectrum means narrow spectrum and broad spectrum. Just mean narrow means to the area or to the spectrum where that number of bacteria they target. Where the broad spectrum, that means they target the lots of bacteria and lots of kind of bacteria to kill them. While always the natural penicillin, these are the acid labile penicillin actually, while uh, the new generation penicillins come that are acid resistant penicillin. Penicillin have not only a problem with acid, it's also have problem with penicillinase enzyme and the lactamase enzyme also. So there is some structures. Penicillin, there is a group drug name. So as we know that lactam ring, thiazolidine ring, acid, carboxylic acid, and there is a acyl amino groups. So there is a R, it means it could be change. So R group, it be if we add this CH2 with benzene ring group, then it if add it here, then it call penicillin G. Then if CH2O benzene ring, then it's called penicillin B. If add this group, there's ampicillin, it's called amoxicillin, and this is methicillin. So as we adding the structure R here, that the name of drug will change automatically. Structure activity relationship. how the structure will change and how the action of drug will happen. So position 1 means sulfur. When the sulfur atom of the thiazolidine ring is oxidized to sulfone or sulfoxide, it improves acid stability but decreases the activity of the agent. For the position second, no substitution allowed at this position. Any change will lower activity. The methyl group are necessary. Position 3, the carboxylic acid of the thiazolidine ring is required for activity. If it is changed to an alcohol or ester, activity is decreased. So not need to change with, you not need to exchange with alcohol or ester groups. Then position 4, nitrogen, it's a must because nitrogen is the part of lactam ring and lactam ring is the main active uh, ring which ultimately show the pharmacological action of penicillin. So in position 5, no substitutions allowed at this position. If no substitution, if any kind of substitution added there, the activity will be decreased or diminished. While position 7 is of course oxygen with double bond. The carbonyl on the beta lactam ring is must, means as nitrogen is must, then carbonyl is must. And then position 6, there is a lot of substitution happened in position 6. In fact, it's a uh, as we seen earlier like that in this position line 6, so a lot of substitution could be added. So position 6 gas substituents are allowed on the side chain of the amide. An electron withdrawing group added to at this position will give the compound better acid stability because these substitutions will make the amide oxygen less nucleophilic, while a bulky group added close to the ring will make the compound more resistant to beta-lactamases. Steric hindrance provide protected to the beta-lactam ring. This is side chain can be replaced with different R group obtain different compound with broad antibacterial spectrum, as this is just a short form of the SAR. And okay, so three chiral three chiral centers are requirement of for penicillin bioactivity. So three chiral center are requirement for at it one who all the at it 1 2 3 5 and 6, 3 5 and 6 are the chiral carbon. There's a side chain called R. Here could be changes happen. Nitrogen group of lactam ring must not be changed. Carbonyl group of lactam must not be changed. The presence of the carboxylic group is requirement of penicillin protein binding penicillin binding protein recognition. Where the esterification of it behaves a prodrug, the viability will be raised. Okay, so the penicillin binding protein is recognized, identified by the COH group. It's if added other group, then it's ester. If then it could behave like it's uh, like prodrug. Especially ester group will what they happen if esterification. Okay, so what are the adverse effect of penicillin? So there is some common adverse effect. The one of main adverse effect of penicillin is hypersensitivity reactions, and it could like anaphylactic shock. So penicillin injection, it's very dangerous sometimes when person have kind of sensitivity with penicillin. If someone patient have a sensitive with penicillin, and unfortunately physician or doctors or any health worker and injected, then definitely it could be lethal to that patient. It's a high sensitivity activity, will active, and this due to shock maybe patient die. So it will be always remember that the penicillin should not use in any patient without testing, without knowing that sensitivity toward penicillin. So some diarrhea, that is watery or bloody, fever, chills, body ache, flu symptoms. Chills, something person have feeling like a cold, urinating less than usual or not at all, severe skin rashes, itching or peeling. The skin rashes is also the part of this hypersensitivity reaction. Agitation, confusion, unusual thoughts or behavior, seizures. It's a very rare. Blackout or convulsion. It's a very rare. Nausea. It's a very common. Stomach pain is also very common. While vomiting sometimes happen, and vaginal itching or discharge. It's also a side effect, and headache. Then the use, what is the therapeutic use of penicillin? So there is a different kind of bacteria where penicillin is used, like pneumococcal infections, that is meningitis, pneumonia, where a streptococcal infection like uh, streptococcal pharyngitis, pneumonia, also arthritis, meningitis, and endocarditis. Staphylococcus infections, meningococcal infections, gonococcal infection, syphilis, diphtheria. Syphilis, it's a kind of disease where penicillin was very common in use. Diphtheria, anthrax, Clostridium infection, surgical procedure in patient with valvular heart disease. So these are the uses of penicillin. So we have learned that beta-lactam, the compound which contains the beta-lactam ring, that ring is 5-membered, 1, 2, 3, and 4. 1, 2, 3, and 4. 4-member ring. That ring is 4-member. One side is nitrogen attached, another carbonyl group is C double bond O. That ring is called beta-lactam. It's a pharmacophore of penicillin. We saw the activity. So finally, thank you all for listening these videos. The next part of this video will consider like this video is only discuss on penicillin, while cephalosporin and rest of all will cover in next videos. So thank you very much for listening us and giving this time. Thank you.